Hi, and welcome to episode 73 of the Breaking Bio podcast. I'm Morgan Jackson, a PhD student at the University of Guelph in Ontario, Canada.
Hi, I'm Tom Housley, a research dog's body at the University of Cambridge in England.
This week we'll be seeing red as well as a lot of other colors with Dr. Nate Morehouse, who's an assistant professor at the University of Pittsburgh. Hi, Nate. You want to tell the listeners a little bit more about your research?
Yeah, great. Well, thanks for having me on. I can't believe this is the 73rd episode. That's quite the accomplishment. Yeah, I'm a biologist at the University of Pittsburgh. I run a fairly large research group here these days. We're 18 people now, 3 PhD students, a postdoc, and a whole army of undergraduates. And the theme of my research for a long time now has been understanding the traits that males and females use to interact with each other. And for me, I have really focused in on a particular interest of my own, which is understanding how animals use color to interact with each other. And that brings our research to a lot of different places. It involves thinking about how animals perceive color, how they produce color on their body surfaces, and how they use that to signal with each other to convey information. And we focus largely on the context of courtship signaling in insects, predominantly butterflies, and now jumping spiders have become a real focus of our research.
Sweet. Well, we got lots to talk about. So when we were doing a little bit of background for this episode, I was going through your CV, and there's a whole lot of stuff of very cool work being done in your lab. And it seems like you've been pretty productive. And now I can see with 18 people in your lab, I kind of understand why that why that's got so much going on. So before we get into the science, what's it like having such a massive lab?
It makes me feel a bit like an octopus with all the tentacles stretched in different directions. So, we have projects that are looking at color polymorphisms in butterflies, and then projects on jumping spider vision and jumping spider signaling. Actually, a big chunk of my lab is studying reproductive protein coevolution between male and female reproductive proteins. So that's forcing me up a very steep learning curve on the molecular side of things. So I'm never bored, ever bored, and I'm oftentimes working long hours, but I love it. It really, we're a bit of a family, as it turns out, with kind of weird siblings and odd cousins and things, of course, but we get along really well. We do, for me, balance in life is really important, and so although we work really long hours, everybody in the lab works long hours, particularly during the summer months when we're rearing large colonies of animals. We try to temper those research hours with some social time as well. So we do go on monthly lab outings where we do something totally unrelated to what we do scientifically. Everything from going to scare houses around Halloween to baseball games. We went fossil hunting this fall, which was pretty awesome. Went to see a roller derby match, and so we end up being really good friends and kind of enjoying each other's company, which I think is critical, especially during those long hours we spend. So, yeah, it's a stretching experience. I'm definitely growing and learning from it myself personally as well.
Yeah. Did you come from a large lab environment while you were doing your graduate degrees?
No. So during my PhD, the lab I was in was fairly small. We, on average, had 2 to 3 graduate students in the lab at any given point in time. So about the same number of graduate students, but not nearly as many undergraduates. As a postdoc, I was at the University de Tours in France, and the French system is structured slightly differently. Here in the U.S., we oftentimes have one PI and then a bunch of more junior people, but in European systems, there's typically a bit more of a hierarchy, so the research groups can be fairly large with a lead professor and then a bunch of younger professors underneath them. So the group I was in France was bigger, but it was bigger because there were more faculty members in the research group. So, yeah.
Sweet. All right, so we'll leave your lab lifestyle alone for now, but we'll get into some of your research. So one of the papers that I figured we should start off with is some of your work on butterflies, and particularly on polymorphisms. Could you explain a little bit about what polymorphisms are and how they're interesting for you, as a color researcher?
Sure. Well, polymorphisms are any time you have multiple phenotypes that are genetically determined within a population, and so that's a really broad category. The most typical polymorphism would be sex. Sex can be thought of as a polymorphism, right? You have different genotypes within the population that produce different phenotypes. When we're thinking about polymorphisms, and in particular thinking about it in butterflies, we're oftentimes considering when you have different color morphs within a population that persist. And these polymorphisms, they're obviously visible parts of the biodiversity around us, but they present something of an evolutionary puzzle, because the implication of a polymorphism is that you have multiple phenotypes, multiple genotypes within a population that have roughly equivalent fitnesses over evolutionary timescales. And that runs counter to the expectation that typically one should just be slightly better than the other and should eventually go to fixation. So it presents a lot of interesting challenges for us in understanding why multiple genotypes with distinct phenotypes are maintained within populations. For us, thinking about coloration, there's been a lot of work on this topic, looking at frequency-dependent selection, for example, and how that may maintain multiple morphs within a population. The particular thing we're focused on right now is understanding the role of the community composition on how individuals interact with polymorphisms. So we're looking at a butterfly species in the group Coleus, where males have to deal with the fact, or have to respond to the fact, that there are multiple different female color morphs in a population. And this is a widespread polymorphism across this group. It's an incredibly high incidence, but it seems to be lost with some frequency. So we've done some looking at the evolutionary history of this. And this particular polymorphism is called the alba polymorphism. And it's named alba because one of the female phenotypes is white, looks albino, and the other is more male-like in coloration. And people have looked at this. They've determined that it's produced by a single locus. It's not on a sex-determining gene, so it's an autosomal locus. It's dominant. So we know a bit about that. We also know that the white female form matures more quickly. It emerges earlier than the non-white female form, and that those females have more eggs mature at eclosion when they arrive at adulthood than the non-white female form. So those white females should be much more profitable for males. They have a larger complement of eggs ready to go right away. They're likely to develop faster. They are able to accomplish this because their wings are white due to the fact that they put less pigment in them. So they seem to redirect pigment from the wings into these important other developmental processes. So really the question is, why all males don't prefer white females, the albomorph? Why isn't the albomorph dominant and end up going to fixation in the population? And as it turns out, some of our early work has shown that one of the clues to why white females may not be the fittest of the two female forms is that white females are easy to confuse with other butterflies that are flying in the environment. So there are other alba females of other species that are very difficult to discriminate from these butterflies, and there are also other white butterflies in the environment. So when we look, quote unquote, through the eyes of these butterflies, we find that males may actually be really confused or may have a difficult time distinguishing these white, potentially more profitable females from other completely unprofitable butterflies in the environment. And that suggests an interesting thing. It suggests that the environment that these animals are situated in, the context of other butterflies around these animals, may actually have fitness consequences for one of the female morphs, and may actually reduce the attention that males give to those females, and may equalize the fitness of those two morphs in a way. But that should be dynamic. It should respond to differences in the co-flying butterfly community and ways that we could predict by going out and actually looking at the communities that these animals are in. And that's a fairly novel concept that we're just beginning to explore now. Are males learning or paying attention to the community of other butterflies around? And is there certainty influenced by the other butterflies around? Is there certainty that these alpha females are a profitable mate option, undermined, for example, by other options that are clearly not reproductively profitable to them?
So how costly is mating for the male butterflies as well then? Presumably there must be some, obviously apart from the cost of missing sort of reproductive opportunities for the first ones, but do they give nuptial gifts or anything like that?
They do. So yeah, there's a couple of sources of cost here. One is just the opportunity cost or the metabolic cost of courting and spending time with the wrong species, for example. But really, we think that the big costs are likely to come from males that actually mate with the wrong species. And that can be really substantial. So these butterflies transfer enormous ejaculates called spermatophores. They contain sperm, but they also contain lots of other things. And when I mean lots in terms of weight as well as diversity. In this species, males can transfer between 8 and 10% of their entire body weight during a single mating.
Wow.
That's huge. You know, for me, that's 18 pounds or something like that in equivalency. So that's huge. Right? That's several bowling balls worth of ejaculate, if you will.
That's pretty much like giving an arm and a leg to your mate.
Yeah. Bizarre to think about it in our own terms. So for these butterflies, that's a huge expenditure. And you certainly wouldn't want to spend that on a totally unprofitable mate, for example. And so we know that males are choosy, actually, and many, if not most butterflies, especially ones where males are making this large investment, males are also selecting amongst mates. And this may lead them to be more wary of a mate option that is uncertain, where they could actually spend this large nuptial gift or spermatophore without any fitness return.
So this talk of sort of the cost of mating leads me to ask you about, you just had a paper published recently about condition-dependent ornaments. I was wondering if you could talk a little bit about that.
Sure, yeah. Well, you know, one of my core interests has been to try to understand these colors in particular, or any kind of reproductive investment, in the context of the life history of the animals that produce them. And there's been a topic that's been of real interest in the field for the past 10 to 15 years, and that is the concept of condition dependence. Condition dependence is the concept that traits, particularly ornamental traits, may be informationally valuable to viewers if they're connected to the underlying pool of resources or the underlying condition of an individual. So they end up being a nice shortcut to understanding differences in individuals and their condition or their quality. And so people have typically looked at this by influencing condition, imposing some kind of stress, like manipulating the resources that an animal has available to it, or making it sick, or otherwise damaging its condition, and then asking whether or not the ornaments respond to that. My interest has been to try to understand how that condition dependence, that linkage between an ornamental trait, for example, and the underlying pool of resources that fund it, might tell us about how that ornamental trait might actually tell us something about the quality of the individual on a whole bunch of different other facets of their individual quality. So to think about this, think about the idea that individuals have a pool of resources to spend, not just on their ornaments, but on other things, on their gametes, on their ejaculate, on their spermatophore, on health, on longevity. And so if we focus on these resources and begin to quantify the ability of individuals to acquire these resources, as well as how they invest them. What is their strategy of investment? When they're low on resources, when they have lots of resources, we can begin to understand how the suite of traits that make up an animal are interacting with each other, and therefore what the likely value is of paying attention to some of these ornamental traits. So my currency of interest has been protein or nitrogen, because for herbivores like butterflies, it's oftentimes quite limiting in diet. And what we found is that many of these butterfly colors that we've been studying are incredibly expensive in this particular currency. So these animals, particularly males, are spending large quantities of a resource that they have in very limiting supply. And what we find is that males that are more resource-rich are more colorful because they have more to spend on these colors. But interestingly, those males also have more to spend on other things. So we just talked about spermatophores quite a bit, and we're just about to submit a manuscript that shows that males that are more colorful produce larger and more protein-rich spermatophores. And the reason for that is that both of those traits are reliant on the underlying pool of protein that those individuals have been able to acquire. So resource-rich individuals have more to put in their colors. They also have more to put in their ejaculates. And as it turns out, we can influence that by challenging them with an immune challenge that draws away protein towards the immune response, and we see that it impacts both the color and the spermatophore size of those males. So it's really that these things are linked because they're pulling off of the same underlying currency. So that's actually been really interesting to begin exploring how traits are all connected when you begin thinking about them in economic terms, really.
Mm. Wow. Yeah, it's really difficult because you can't think of any of these traits really in isolation. You have to think about how they affect everything. So I think one of the other things that I was going to ask, and going back to what you were talking about with the white butterflies, I saw that you've been working on some where the Pieris rapi, where to our eyes, the males and females both look white, but to the eyes of each other and to their predators, then they look quite different.
Yeah, well, so one of the fallacies that thankfully the field of animal behavior got over 20 or 30 years ago was the idea that our eyes were good surrogates for the eyes of animals, that the things that we could see were the things that most other animals saw in the world around us. And one of the things that exploded that concept for us was that we're a bit of an anomaly in not being able to see ultraviolet. So UV wavelengths are things that are visible as colors to a wide variety of animals: lizards, fish, birds, and butterflies, most insects. And so people always kind of laugh when I give presentations because I say I'm studying color, and here's one of the animals that we study color in, and then I show them a black and white butterfly. And they think, why the heck would you pick the cabbage white? Well, it turns out that those butterflies absorb very strongly in the UV in parts of their wings, the white parts of their wings actually. And so what they end up having is a big contrast between absorbing strongly in the UV and reflecting strongly in the wavelengths that we can see. That's why they look white. And that's really the basis for color, is big differences in the amount of light that come off of a surface at different wavelengths. And so animals that can see UV as well as the wavelengths we can, like these butterflies are able to, see that contrast, the strong absorbance in the UV, strong reflectance in other wavelengths, as being a really vivid color, much like a really vivid orange or yellow might appear to our visual system. Turns out females pay attention to that in the cabbage white, and they prefer more colorful males as mates. But it also turns out that those more colorful males are more obvious to the birds that feed on them. So there's some debate about how much birds feed on these butterflies. We know they do. We know that they're not their favorite thing to eat, but we know that they'll eat them when they're hungry. And so it's possible that these more attractive, more colorful males are running a higher risk of being picked off by birds, which is kind of an interesting idea.
Well, it all goes to show just how freaking complicated the natural world is, and how there are so many things going on. So one of the questions, so the butterflies that you're focusing on, the alba ones, Coleus or Cleus?
Yep, Coleus, yep.
Coleus. Are those North American native or European native?
They're North American native. We are studying two co-occurring Coleus species, Coleus eurythemi and Coleus phallotici, and their ranges have changed, though, within recent memory, largely due to the cultivation of alfalfa, which is one of their main host plants. So we cultivate alfalfa as a feed crop for cattle, and also as a nitrogen-fixing crop that gets tilled into the soil to augment the soil nitrogen. And these butterflies have taken advantage of our cultivation of those crops, and that's actually led to increased range overlap between the two and increased hybridization between these two species, which is kind of an interesting set of questions in and of itself.
Cool. So the question that I have is, you're comparing it to the cabbage white, which, correct me if I'm wrong, but is a European species that's been introduced to North America in the relative recent history, right? So
Yeah.
are these conflicts that these sexual interference, reproductive interferences, are these new things that that the native butterflies are having to deal with now that there's a lookalike on the scene?
Well, that's a good question. So it's certainly the case that it is a much bigger issue for them now, because Pyrus rabii, the cabbage white, it was introduced in 1860 or 1861 on the East Coast and then slightly later on the West Coast, and is an incredibly successful invasive butterfly. So the populations of it are much larger than white butterfly populations might have been historically in any of the range of these Coleus butterflies. However, there are a number of native white butterfly species that would have co-occurred and still do co-occur. So we have a number of Pyrus species that are native to the U.S. that were not introduced, including some that are now threatened. Pyrus virginiensis is a particular example of that on the East Coast. There's also another genus on the West Coast called Pontia protodici being a very abundant black and white butterfly in those environments. And the colors on their wings are equivalently overlapping with these alba females. So I would say that what we're looking at is likely to have been important in the evolutionary history of these animals, but it's a particularly acute issue for them now.
Right. So they may have had to have been dealing with it for the last million years, but it's becoming a much more common factor for their survival or their mating chances now that you've got something that is everywhere. As people I'm sure have seen, North America have seen the cabbage white. It's the most common butterfly you'll probably see in North America at this point.
Anywhere, really.
Yeah.
Pretty much everywhere in the world, it's one of the most common butterflies. Yeah.
Yeah. Cool. So maybe we should move on to some of your other work now. So staying with the colored theme and how it factors into life and death struggles. You've got some work on jumping spiders recently published, which is showing that they have color preferences for their prey. And to do this, you were playing around with some crickets and feeding the crickets different colored dyes, and then feeding the psychedelically dyed crickets to jumping spiders. Can you explain a little bit about what that resulted in?
Yeah, sure, absolutely. Well, in going into this, we did a little bit of background research. The paradigm has always been that animals that use color to defend themselves, for example, to show that they're distasteful, are doing so with a particular audience in mind, and that audience has always been birds. In the mind of the researchers studying this. Birds are out there, they're pecking away at things, they're avoiding the red and black stuff when they learn that it doesn't taste very good. And what we discovered was that there's really almost no equivalent literature for predators that are not birds, in particular for predators that are invertebrate predators, so things like predatory insects or predatory spiders. So we were really curious about whether or not spiders that appear to use color in other contexts, so the spiders that we use color in courtship, and I can talk about that in a second, whether or not they were making any decisions based on the color of their prey. And that would be interesting because the dynamics between tiny little predators and tiny little prey might be totally different than they would be between birds and larger prey animals. So yeah, we took these colorful jumping spiders from the American Southwest. We know their prey communities are full of differently colored prey items. Some of them are chemically defended. The ones that are chemically defended have a tendency to be red, orange, or yellow. So we walked into this thinking, well, if we were a jumping spider and we were deciding amongst prey items, we would probably avoid those colors. And so yeah, we took advantage of the fact that the teeny tiniest crickets that you can get, the ones that come out right from the egg, are translucent enough that if you feed them colored water, they'll uptake it into their gut, and it actually changes their body color. So we made this kind of cricket rainbow, and then we introduced this set of colored crickets into an arena, and we monitored what jumping spiders attacked. What did they attack first? What did they attack most? What did they attack least? And we did this with males and females and also with juvenile jumping spiders. And it turns out that no matter what the jumping spider was, if it was a male or a female or a juvenile, that they attacked red and yellow prey items at lower frequencies than they did other colors. And in particular, they attacked them much less than they did blue-colored crickets. The other thing that we found was that these were animals we caught in the wild. We brought them right back in, and their responses to prey are likely to be a mix of anything that they are born with, essentially, innate color preferences, and anything they've learned by dealing with prey in the field. And so what we found was that when we first tested them directly out of the field, they avoided these red and yellow prey items, but then after a couple of weeks, that bias declined, suggesting that some of it may be learned.
So they didn't get sick when they ate the red and yellow ones, so they didn't care if they weren't put off by them later on.
Well, yeah, this is always an issue for us because these animals are quite clever, even though they're really tiny. And so they can actually learn in the context of these trials. So that was always an issue for us. We were always feeding them uncolored prey items, white-eyed fruit flies, on the off-season, as it were, trying to avoid manipulating their prey preferences. We have just submitted another paper that shows that they can actually learn using red. So we can train them to avoid red prey items, or we can train them to prefer red prey items. And we do that using a milkweed bug that is chemically defended when it's fed, when it's raised on milkweed seeds, but it's completely delicious, at least to the spiders, when you feed it on sunflower seeds, and they'll grow up on sunflower seeds as well. And in comparison to a control group, we found really strong aversive learning in the group where the red prey items were chemically defended, and pretty strong preferential learning when they were fed the red prey items that were tasty. And that they actually transfer this. So not only do they change the way they interact with these milkweed bugs, but they change the way that they interact with colored crickets, which are a totally different body shape, and they smell different and they act differently. And so these seem to be more generalized learning preferences based on red.
That's pretty interesting. So we're going to jump back to the blue thing a little bit because I find this really interesting. So blue is very uncommon in the animal kingdom. Like the pigments itself I think are very expensive physiologically to produce. So you may have structural colors coming through where the actual physical shape of the insect is reflecting blue. But the pigment itself is quite uncommon and hard to make. Even though it would stand out fairly well for mating displays and stuff. Do you think that this increased predation on blue is also helping beat down on animals and investing in this expensive property?
Huh. Well, that's an interesting question. Yeah, so blue pigments are rare. They're bile pigments that are typically the ones used by animals to produce blue. So biliverdin and other bile pigments, porphyrins as well. They're rare, and you're right that it's typically structural color that is used. We interpreted this result as a potential sign that these animals are exploring a novel color, that they don't see it very often, and that they're actually much more likely to attack it because they are interested in evaluating whether or not it's a valuable prey item. So it's something called neophilia. We do see this in the animal kingdom, that really novel stimuli oftentimes produce exaggerated exploratory behaviors towards those types of stimuli. But we really don't know. I do think that it would be a barrier for animals that would use blue, when blue is a very rare color in the environment, because they might be subject to these exploratory attacks by things like jumping spiders. So it's an interesting question. We don't see blue very much in their prey community, and so we don't really know if that's driven by this kind of behavior, or if it's just a consequence of its general rarity in natural environments.
Cool. So we're getting pretty close to the end of time, but we need to touch on some work that your students, or rather your postdoc, is wanting to do and is looking for help from the public to make happen. So you guys are currently crowdfunding a project that's going to be looking at the role of color. I'm pretty sure this is getting the importance and the role of color in mating in salticids, in jumping spiders, rather than in prey selection. Can you tell us a little bit about what you're hoping to do and how you're hoping to pay for it?
Sure. Yeah, absolutely. So in the process of doing this work and realizing these animals are really responsive to color, we were kind of persistently irritated by the fact that although they behave in response to color, we really knew nothing about how they saw color. And in fact, if you were to look at any closely related spider, or even distantly related spider, what you would find is that these animals don't have what we call true color vision. They have a system that's based on two different sensitive color-sensitive cells in their eyes. They have a UV-sensitive photoreceptor, or color-sensitive cell, and a green one. And that result is robust across a wide range of spiders, both in the jumping spiders as well as in wolf spiders and crab spiders. Seems to be a common feature of spiders. That would give them some limited ability to discriminate colors between UV and green. So blues and violets and things they might be able to see, but they wouldn't allow them to see red or yellow at all. And that stood really in stark contrast for us with the behavioral evidence that we had in hand. We also know that females are paying attention to red ornaments in males in the context of courtship. Males with red faces do better in courtship than males that have that painted off. And so we spent some time trying to figure out how these animals see, and what we found was that they have UV and green sensitive cells in their retinas. And that was actually really discouraging to us, because it didn't solve the mystery. We were doing this under dim red light to try to avoid bleaching the retinas as we were measuring them. And one day, our collaborator Tom Cronin flicked on the white lights just to look at a section of the retina before putting it away. And he saw something which actually turns out to be this very serendipitous key to this whole thing. What he saw was that within the retina, there is a red filter. And that red filter sits right over a set of green-sensitive cells in the eye. And what it does is it only allows red light to hit those green-sensitive cells. It essentially turns them into red-sensitive cells. And so they achieve trichromacy, like we have, using this filter, and that opens up their ability to see red and orange and yellow and make discriminations between those colors in green. What we're tackling now, so we're just about to send that piece of the pie out for a review. That's really exciting. First clear evidence of true color vision in spiders, at least from the level of the retina. So that helps us to understand why they're responsive to color. What's interesting is that when we saw that, we said, oh, huh, interesting. How many spiders have this? We began sampling spiders across the family tree of spiders, if you will. And what we found is that it seems to be the case that that red filter that gives them color vision is restricted to the group of colorful jumping spiders that most people are seeing on YouTube these days. So the group that we're studying, these Habernatus jumping spiders here in North America. There's another group in Australia called the Morehouse jumping spiders, or peacock jumping spiders. And so what we think occurred here, what we're trying to test, is number one, that this red filter appeared before this crazy diversity of color in courtship arose in these jumping spiders. It's likely to have had immediate benefits for them in choosing amongst prey items. So we want to see exactly when this filter arose, but then we're also interested in whether or not, once they saw color, there was selection for the use of those colors in courtship. So without this true color vision, some colors like reds, oranges, and yellows would be totally meaningless. They wouldn't be seen by the female that was evaluating a male that was displaying, for example. Once you have the ability to see that, this opens up this whole world of color for these animals, and we'd like to see whether or not evolution, in a sense, populated that new world of color with male ornaments. So because there was meaning to these new colors, males were able to exploit that new area of color space, really, and that led to some of the rapid diversification of male color in these groups. So it may really help us to understand how spiders went from largely drab to suddenly colorful and diverse in this group of animals. And it may just simply be that they couldn't see the colors before, and that as soon as they could see the colors, this whole world of color opened up for these animals, and they were able to begin using color in their courtship rituals.
It's like the Wizard of Oz for jumping spiders.
Yeah, yeah. So we have some footage of this stuff. I'll just do a quick shameless plug for this. There's two places people could go if they're curious about seeing what we're talking about, because these animals are just incredibly compelling. One is that we have some videos up on YouTube, and they should look for either jumping spider courtship in 4K. So the camera enthusiasts out there might be really excited about the fact that we have an incredible set of sequences of jumping spiders courting each other in 4K video. Or they can also look for my postdoc, Daniel Zurich, who's been leading a lot of this research in my lab, and he has his own YouTube channel. The other place people can look for some additional footage and information is that we do have a crowdfunding site up at experiment.com. And if they go to experiment.com/projects/why are some jumping spiders so colorful, with hyphens between all of those, I imagine you'll put this up in the notes for the audio folks.
Yeah, we'll definitely put links to these on our website at breakingbio.com for anybody who's listening. You can find them there. So don't worry. We will have links available to you so you can go find out more and see some of these crazy videos.
Yeah, we've got some video up there of us in the field and some explanation of what these animals are doing, some of their dance moves and stuff. So it's a pretty fun site to go explore.
Cool. So what is the crowdfunding? You mentioned it briefly, but what are you guys hoping to do with the money that you raise through experiment.com?
Well, so it's really largely to fund an expedition to the American Southwest and West Coast to collect a bunch of species of these jumping spiders to get a better understanding of when this red filter arose that gave them color vision, and then to also begin measuring the males, these colorful males in these different species, to ask whether or not before this color vision arose, is everybody drab, and then afterwards you see this proliferation of color in the wavelengths of the colors that they could now see. And so we're really just hoping to go spend a couple of weeks on the West Coast in the late spring collecting these animals in the field. And people that support us on this get updates from us. We're going to have a video diary in the field. These environments are just really wild and beautiful. Ocotillo forests and desert canyons and, you know, brave rattlesnake bites and peccary attacks and things out there. So it's fun. It's fun field work to go catching these things.
Awesome. Well,
Yeah.
I think we're pretty much out of time, so we're going to encourage people to go and help get you guys into the field and get some of this research done, as well as get some of these outreach pieces of media out there as well, because I'm sure they're going to be fun to watch as well. But where can people follow along with all the other stuff that you and your lab is working on currently?
Well, we're about to put our own website up with blog posts and other multimedia, and that will be at morehouselab.pitt.edu. And so stay tuned for that. That should be up not too long from now. I also have my own personal website where some of this stuff goes up, and that is biology.pitt.edu/person/Nathan-Morehouse. My last name is Morehouse. So those are a couple of places. Watch our YouTube channel. We're putting a bunch of stuff up there. We're also tweeting from Morehouse Lab as our Twitter handle, and mine is Zap Kapow, and so people can keep track of our exploits through that medium as well.
Awesome. And like we said, we'll have links to all of these various sites, as well as to Nate's Twitter handles and various other things. You're going to have to ask him yourself where Zap Kapow came from. We'll leave that up for the Twitter explanation. Maybe he's a Batman fan. Like I said, you can find all those links and much more, including past episodes of our podcast at breakingbio.com. You can contact us on Twitter by following us at Breaking Bio, and you can also find us on iTunes and Facebook by searching for the Breaking Bio podcast. I'd like to really thank Nate for joining us this week, and we'll hope you all listen again next week.
Thanks for having me, and I also want to thank Dr. Lisa Taylor and Dr. Daniel Zurich, as well as Sebastian Echeverri, the people who have been really critical in some of the work I talked about at the very end, and Lisa Lemary on the Coleus stuff. So thanks, guys. Great to talk with you.